Okay, so I would be talking about some algorithms that we developed to predict drug resistance in drug targets. So before I go ahead, I just wanted to briefly overview how a competitive inhibitor actually works. So for instance, here what I have on the farther side is a drug target, the HIV protease protein, and it's bound to its native substrate, the peptide ligand, and that's the natural state. On the other hand, on this side, we can see the same drug target, the HIV protease, it's bound to ritonavir, which is a protease inhibitor drug. And because it's bound to ritonavir, here, of course, it no longer binds its native substrate, the function is inhibited, and the drug is supposed to work. However, of course, mutations in this drug target protein can impair this drug binding, prohibit the binding, and this can lead to drug resistance. So what we wanted to do was we wanted to develop an efficient approach that was structure-based, that could predict drug resistance, that could predict these resistance-causing mutations a priori in drug targets, so before they're seen in clinical practice. We also wanted to make sure that the approach is applicable to a generic drug target system. So given any drug target protein and a drug, we should be able to use our approach. And it should not rely on existing training data. So we should not need to know too much about what kind of mutations exist out there for the drug target. So because of that, it should be applicable to emerging diseases as well as new drug targets. So since I'm talking about searching for these resistance-causing mutations, I wanted to show what exactly are we looking for. So before we get there, here there's a pseudograph, I would say. So on the y-axis is the binding energy, and the first column, which is the wild-type protein, we can see that the drug is actually a better binder for the wild-type protein compared to its native substrate, which means that the wild-type is actually sensitive to the drug, so it's inhibited. On the other hand, if you look at, for instance, mutant 2, you can see that because of the mutation, that binding profile is switched. And the drug binding is impaired, whereas the native substrate binding is impaired as well, though it's close in range. So there are two things that we are looking for. The first one is, of course, that if a mutation is supposed to be resistance-causing, the mutant should impair drug binding. And the second one is a little more subtle, which is that it should also maintain near wild-type binding to the native substrate so that it can still perform its original function. So, since we'll be searching for these mutants, the question is, well, how big is the search space? And as usual, the easy answer is, well, it's exponential. But the idea here is that if you look at the sequence space, let's say, for an active site of just 10 residues, at each position you allow 20 amino acids, even in terms of single mutant sequences, we are looking at 200 mutation sequences. It becomes really complicated, however, when we look at the associated structures, because of course, for each one of these mutation sequences, there are associated mutant structures. And those are exponential in nature. Just to illustrate the point, for instance, on top, we can see it's a zoomed-in version of an active site. It's the same sequence, so the phenylalanine side chain is displayed. And we can see that in the first case, because of a different structural conformation, there's a steric clash with the drug. So that particular structure is going to give us really bad binding energy. On the other hand, when we look on this side, due to a different structural conformation of phenylalanine, it's a much better structure. So it's actually a search in the structural conformations. So what do? We use an efficient protein redesign algorithm to search through this search space and give us the top-ranking substrate binding mutant structures. So these are all the mutations, all the mutant structures that bind that bind the native substrate optimally. And how it does that is that given any input protein structure, it quickly eliminates all the unfavorable solutions. For instance, those steric clashes and other ones that it can guarantee are not part of your optimal structures. And it's worth mentioning that for most active sites, on a single processor, it runs anywhere from a few minutes to a few hours in order to generate those top mutants. So in order to use it for resistance search, what we do is we start from the wild-type structure and the native substrate. We perform an RDE search on it, and what we get is we get a list of mutants that actually maintain near-native binding. For each one of these near-native binders, then, we have to score it for drug binding because we want to make sure that it actually does impair drug binding. And at that point, we evaluate a score. And what we are looking for is that for any given mutant, if the drug binding is impaired more than the substrate binding, then it's a good enough candidate for resistance at this stage. So for each one of these mutants, two things are evaluated. It's substrate binding as well as its drug binding. So after we're done the first stage, what we want to do is we want to add a more biophysically accurate yet more time-consuming second stage that can fine-tune these predictions from the first RDE stage. And at this point, we use MMPBSA, which is and we use the Amber implementation of this particular algorithm. And what's worth mentioning is that we cannot actually use this right away because for each one of the target structures, MMPBSA takes anywhere between 30 minutes to an hour to run. So if you have exponential number of structures, that won't be possible. So in order to do this, for every one of the resistance-causing mutations that comes out of the first stage, we evaluate its binding energy again using MMPBSA this time. And we use, of course, we evaluate it with the native substrate as well as with the drug. The score is computed again. If we still find that the mutant does impair drug binding more than it impairs the native substrate binding, it's flagged as resistant. We applied our model on 3 different systems. There was isoniazid and ACP reductase, which is for tuberculosis. There was glycocanadial kinase, and there was ritonavir and HMB protease. And what we find here is that for isoniazid, for instance, there are 5 well-known mutations, and 4 of them are actually recovered by our algorithm. I again want to remind you that the algorithm actually works without any knowledge of existing resistance, so it does not know that 5 exist out there. The second one is for glycocanadial kinase. There are 2 known mutations, and both of them are recovered. At this point, we also performed an additional test, which was for a new inhibitor called ponatinib, which is supposed to be a replacement for Gleevec, because it targets the T315I mutant. And what we found was that if we apply our approach to ponatinib at ABL kinase, we actually do find that T315I is indeed sensitive to ponatinib, as expected. Similar results were also obtained for HIV protease, and what we find is that along with the major PI mutations that are known, our method also recovers the rare resistance-causing mutations, such as BH2D. And at the end, what I want to add is that another good thing about using a structural approach, other than the fact that we don't need to rely on existing training data, is that for each one of the mutant structures, for each one of the mutations that it predicts as resistance, there's a potential cause attributed to it. Because it generates a structure at the end, you can look at that structure and figure out why something is flagged as resistant. So for instance, in this case, we're looking at it's a zoomed in version of an HIV protease double mutant B82A I84V. It's known to be resistant causing, and we're looking at 2 superimposed structures. The green one is the wild type, and there's the drug. It's ritonavir, and the blue one is the mutant structure. And what happens here is that there's a loss of metabolic contacts with the drug that impairs drug binding when the mutation occurs, and that's the cause of resistance. And all of our methods and their results were published last year. And at this point, I'd like to thank everyone in the MOST lab and Professor I. Lillian and everyone in the Lillian lab as well. And if there are any questions, thank you.
Are there any questions for Maria? With the secondary drug
Yeah.
are you going to be looking at resistance possibility?
Yeah, so we actually had a list of candidate mutations that we think are sensitive to statin, and we published statin unless they appear to be different, so you don't know what validated. But that particular one was indeed statin sensitive because it is actually geared towards statin.
Right.
What was the correlation in the score from the first round to the second round?
Correlation, sorry, what's
In the score for the dead-end elimination versus...
Oh, these two actually use different different energy functions. The MMPBSA one is a much better energy function, if you will. The DEE actually uses a pairwise function to quickly eliminate everything else. So we never actually looked at correlation, but there's a significant reduction. So for instance, if you're looking at about 200 structures coming out of RDE, at the end when you score them with PBSA, you might take 40 to 50 of them because it allows more flexibility at additional cost, computational cost.